This particular challenge is concerned with jumping across biomedical contexts. So what do I first mean by heterogeneous associations? So mining large-scale heterogeneous biomedical data sets often involves analyzing a large number of various relational data sets. And a natural representation of relational data sets are graphs. So here in this slide, I show 5 different networks that capture different types of associations. So there on the left side is a network between molecular functions, then the network of diseases that relates diseases to each other, or disease symptoms. Then perhaps a canonical example would be a network between genes, various types of genetic interaction, physical protein interaction data sets, or perhaps some more recent data sets that describe the interplay between environmental factors. So just to ease our representation, I will represent each network with a single node, with a blob of the respective color, and with a loop that indicates that behind this node there is actually a network that relates all objects of this particular type, such as molecular functions, diseases, and symptoms. Of course, in order to do integrative analysis and to make predictions that span across different data types, we cannot treat these data sets in isolation, but we need connections and associations that relate these heterogeneous types to each other. So one way of how we can connect these nodes is actually by introducing these cross-domain associations. For example, between genes and gene ontology terms, there's an edge which indicates there is a data set behind this edge that relates genes to gene ontology terms. And as the previous speaker has mentioned, this could be annotations from either manually created or data-driven ontologies. In terms of an edge between genes and diseases, there's a natural representation of a data set which contains gene-disease associations, or between diseases and disease symptoms, there's a disease symptom data set. Of course, we can include many more entities. For example, here I showed chemicals, pathways, molecular signatures, and altogether there are 16 edges in this graph, and meaning there are 16 data sets that we will model in this particular example that is shown here. Now, what's an interesting question that we can ask once we have this complex data system? We would like to ask some queries and would like to query the system and get some predictions about it. For example, if we are interested in understanding better what are genetic underpinnings under various disease symptoms, we may want to profile genes in the space of disease symptoms. So how can this be done? As is shown here on the slide, there is one way that we can relate genes and disease symptoms, and this is through diseases. So we could go through disease associations and then through clinical manifestations of diseases via symptoms. But that's perhaps the most obvious way, but it's not the only way. So another way would be to first profile genes in the chemical space, then profile chemicals in the pathway space, then pathways in disease space, and then from this connect diseases to disease symptoms. And there are many more. So our primary observation is that different ways of relating objects in a fusion graph, such as the one shown in, here, carry different semantic meanings. And this means that depending on a concrete application and concrete semantic that we consider in our experiments, we can get different results. And our claim is that mixing, discarding, or ignoring different semantic can not only reduce predictive performance of the models, but also affects their explanatory capabilities. So overall, what I would like to convey or present in this talk is propose an approach towards better understanding of heterogeneous associations by connecting different biomedical contexts. In terms of prediction, the questions that we are asking are, can we estimate whether a given gene and diseases are likely to be associated, and beyond going beyond pairwise associations, can we find a set of genes that taken together appears most significant with respect to another set of genes? And I will formalize this in a bit. In terms of understanding, we would like to better understand what are the different similarities between objects that are implied if we take if we consider different ways of connecting these object types in the collection of data sets that we consider, and how we can help users to choose or combine appropriate statistics in a concrete application. Let me introduce our modeling approach, and before that, a bit of terminology. So I will use the term size K module to refer to a set of K objects that have a specific property that is shared among all members of the module. Then candidates is a term which I will use to refer to all objects that are potential members of the module that we would like to infer. And pivots are objects that we consider to assess the significance of a particular model. So the particular goal that we are trying to solve, or the problem that we are tackling, is we would like to find a size K module of candidates that is maximally significant with respect to a given set of pivots. So this is the problem that we are trying to solve. So a bit of formality, let's assume that we have a set V, which is a set of all the candidates, and we have some function, which is a set function, and it's a module scoring function. So function F takes as its input a set of candidates and produces a non-negative real value, which indicates the quality of the quality of potential module. So having defined V and F, this leads to the following optimization problem. We would like to find a set X star such that the set, or this module has the best quality in terms of function F. And by the best, I mean the best over all possible sets of size K. Now, if F is not a trivial function, then this is a quite difficult problem, because in a way to get an optimal solution, what we would need to do is actually exhaustively enumerate all sets of size K, and this is not feasible. So in order to still be able to provide good predictions with some theoretical guarantees, we need to make some assumptions about the functions F. And in particular, the assumption that we make is that function F satisfies the property of diminishing returns. So what I mean by the property of diminishing returns, I will illustrate with this following example. So suppose we have a set of candidates, and this set contains eight objects. Just for ease of representation, let's say that there are 8 genes G1 to G8. And we have a function F, and for one reason, we would like to evaluate the utility of 4 genes G5 to G8, so this set of 4 genes, and measure how good module do they represent with respect to some pivots, which are not shown here. Now, the idea that the function F must satisfy diminishing returns means the following. For example, when we add gene G6 to a current module, then the incremental gain of adding G6 to the current module decreases when the background in which gene G6 is considered increases. So intuitively, this means that the utility of G6 is the greatest, is greatest where, pardon. The utility of gene G6 is greatest when gene is actually incorporated into the model. And the implications of the diminishing returns or diminishing marginal gain is that when we infer the module, then at each step, the relevance, the significance of a particular gene is discounted based on how much of its representation is already captured in the module. This means that as the module grows, this particular candidate G6 is added to the module at the point where it provides the most useful insight to the module. And if it would be included at any later stage, then its contribution would be only smaller. So formally, what I described is something that is known in combinatorial optimization as F being a submodular function. So why is this a very useful property? and not only mathematically elegance is the fact that if f is submodular function and there are some other technical constraints that are satisfied, then we have very efficient and fast algorithms which allow us to quickly find modules of size k that are good in their qualities. And in particular, a greedy search algorithm here has a worst case guarantee of being 1 minus 1 over e approximation to the optimal solution. And if you recall, the optimal solution would be, given f, would be the one that would go through all sets of size k. So I mentioned that the greedy algorithm is the one that provides us some theoretical guarantees, and we will be using it. So the model that we use consists of the following 3 main steps. So the first step is to derive a semantic based on a concrete collection of heterogeneous data sets that we have. Then in the second step, we start with an empty module, and then we perform k iterations if the output is a module of size k, and at each iteration we score the candidates based on module scoring function f, which we call concentration significance, and then we pull into the module the most promising candidate. So there are 2 crucial parts or 2 crucial ingredients to this algorithm, and the first is obviously how will we estimate the semantic of interests. And the second one is what is actually this function concentration significance, what is its form. So for the first part, we estimate semantic using a latent variable model, and in particular a collective latent variable model, which is able to take a collection of heterogeneous data sets, such as the one I shown in the beginning, and can cofactorize all of the matrices simultaneously. So the matrices represent data sets. And inferring this latent space, we are then able to actually walk in the latent space and derive all the profiles of the objects that we have in the system. So in this way, we can actually capture any semantic, or we can derive any semantic that can be described within the system of the data sets that we have. And for the second part is the module scoring function, and intuitively I will say that the module scoring function consists of 2 parts. And the first part is the part that evaluates the relevance of the current model with respect to the pivot set. And the second part is the part that evaluates the diversity of the model or the coverage of the model. And here diversity and coverage really depends on 2 operating regimes which are detailed in the paper. So in terms of experimental validation, I will show 2 use cases. And the first use case is the canonical example of predicting gene-disease associations. And for this task, what we consider is the exact fusion scheme that I outlined in the beginning of the talk. This scheme contains 16 edges, which are 16 data sets, and 30 nodes, meaning 13 types of objects. And we took special care to try to prevent circularity in the data and contamination with known gene disease association. So our setup is the following. For each disease in the corpus, and we considered around 300 complex diseases, we do leave 1 gene out, cross-validation, and use the remaining genes as pivots. And then we report the area under the ROC curve and area under precision recall curve as a measure of the quality of the model. So what are the semantics that we consider? So we wanted to study various kinds of semantics. In particular, these are a few of them that we considered in the experiments. So for example, the first one is the one that relates genes, which is G, to chemicals, and then diseases, and then disease symptoms. So the path in the graph that corresponds to this particular semantic is the following. So genes to chemicals, to diseases and disease symptoms. Whereas the second semantic is really simple. It's a direct semantic between genes and chemicals. So we derive these semantics based on the latent variable model that we consider. So how the results look like? So the set of box plots that are shown here show the area under precision recall curves for various semantics. So on the x-axis is the area under precision recall curve. On the y-axis are the semantics which we have seen in the previous slide. And each dot here in the box plot corresponds to 1 disease. So remember that we had more than 300 of them. So what are the few observations that we can see here? So first, you can see, or you were able to see that performance varies substantially across semantics. And this provides some insights into the utility of semantics and already hints that for different applications or for different diseases, different semantics will provide will be more useful. And then another observation is that the last predictor, which is predictor that combines different types of semantics in a principled manner, and which we described in the paper, is the one that typically performs better for most of the diseases, but not all of them. Now, gene disease associations are actually a very trivial example of modules, because these are modules of size, of size 1. What we would like to test is, or provide some insights about into the method when actually detecting disease modules of size of larger size that goes beyond pairwise relationships. And for this study, we consider the same set of 16 data sets, and the setup is similar as in the gene disease association prediction task. So the data is similar, but the setup is a bit different. And in particular, for a given disease module, we randomly remove a fraction of disease genes from the module, and then use remaining genes as pivots. And our goal is to detect size k modules where k is the size of the full module. So then we study various metrics on the results, but what I will report here and show are the recalls values. So this particular heat map shows achieved recall values which are stratified by different disease classes. So what we can do is map around 300 diseases onto disease ontology, and these are the disease classes that we get. And here on the x-axis, we can see these are different semantics that we consider in the experiment. So the first observation is that there is some, as you can see from the colors, there's quite some difference in the utility and performance of different semantics. Overall, it seems that the less semantic, which is the one that combines different individual semantics, performs best. Additionally, what is interesting is that related diseases, such as diseases that are related to immune systems or that belong to the same class, generally have similar patterns of recall. And this is something that would be expected of a method that makes robust predictions. And furthermore, another interesting result is that highest true positive rates are usually achieved in the early iterations of the algorithm, which make the method appropriate for doing tasks such as gene prioritization. So to conclude, what I described is a method for module detection that can be used beyond detecting disease modules or predicting gene disease associations for modeling various types of objects that we have in the systems, and actually to connect objects that are not directly related through different ways. There are two key ingredients to the method, and the first one is collective latent factor model, and this is important because it allows us to derive these different ways of relating objects of different types. And the second important concept is submodularity, which admits some theoretical guarantees about the quality of the modules that we find. So with that, I conclude. I'm happy to take some questions.